Of course, here's a great, deep, detailed summary, but still in simple, clear language, pulling everything together so you can see the big picture of eukaryotic cells and what makes them special. What are eukaryotic cells? Eukaryotic cells are big, complex cells with many specialized parts inside them. They are found in protists, tiny, mostly water-dwelling organisms, plants, animals, and fungi. Basically all living things you can see without a microscope, and many you can't. The word eukaryotic comes from Greek, meaning true nucleus, because these cells have a real organized nucleus. Nucleus, the brain of the cell. The nucleus is like the brain or control center. It holds the DNA, which is the cell's instruction manual. The nuclear envelope, a double-layered membrane, surrounds the nucleus, protecting the DNA inside. DNA is neatly packaged into chromosomes so it doesn't get tangled. Compartments inside the cell. The inside of a eukaryotic cell is divided into many compartments, like rooms in a house or offices in a building. These compartments are organelles, little organs of the cell, and each one has a special job. Organelles are surrounded by membranes, thin walls that keep them separate and organized. Organelles give the cell power, structure, and order. Some key organelles include mitochondria, make energy like power plants, ribosomes like factories, endoplasmic reticulum, ER, transports and makes proteins, lipids like conveyor belts, Golgi apparatus, packages and ships proteins like a post office, lysosomes break down waste like recycling centers, chloroplasts in plant cells make food from sunlight like solar panels. Cytoskeleton, the cell's bones. The cytoskeleton is a network of tiny protein fibers inside the cell. It gives the cell shape, strength, and helps it move things around. It's like the steel frame of a building or the bones of your body. Movement, locomotory structures. Some eukaryotic cells can move around thanks to special structures. Cilia, tiny hairs that beat like oars on a boat. Flagella, a long tail that whips back and forth like a sperm cell's tail. Differences between plant and animal cells. Plant cells, like those in trees or flowers, cell walls, hard outer layer that gives the plant strength and shape, like a box. Plastids, especially chloroplasts, make food from sunlight by photosynthesis. Large central vacuole, big bag storing water, nutrients and waste, helps plants stay stiff. Animal cells, like those in humans or dogs, have no cell walls, making them soft and flexible. Centrioles help animal cells divide during reproduction. These are almost always missing in plant cells. DNA and chromosomes. DNA holds all the instructions a cell needs to grow, work, and reproduce. In eukaryotes, DNA is organized into chromosomes inside the nucleus, making it easy to copy and share genetic information when cells divide. Think of a eukaryotic cell like a city. Nucleus, city hall, makes decisions. Mitochondria, power plants. Ribosomes, factories. Golgi apparatus, post office or shipping center. Cytoskeleton, roads and bridges. Cell, city border. Organelles, different buildings. Cytoplasm, air and space inside the city. Main takeaways. Eukaryotic cells are big, organized and powerful, full of specialized parts working together like a tiny city. Their nucleus and organelles make them very different from simpler prokaryotic cells, like bacteria, which don't have these parts. Plants and animals are both eukaryotic, but have big differences in what their cells contain. This summary gives you a full, deep and clear view of what eukaryotic cells are, how they're structured, what makes them special, and how plant and animal cells differ, all in simple, easy to understand words. Would you like me to make a mind map or diagram of this? Of course, here's a deep, detailed, yet still easy to understand summary of everything about the cell membrane, capturing every important point clearly and simply, but with thorough details so you fully grasp it. What is the cell membrane? The cell membrane, also called the plasma membrane, is a thin, flexible layer that surrounds every living cell. It separates the inside of the cell from the outside world and controls what goes in and out of the cell. Without it, a cell would fall apart or get destroyed by harmful things outside. Discovery of the membrane structure. Before the 1950s, scientists didn't know exactly what the membrane looked like because microscopes weren't powerful enough. In the 1950s, the invention of the electron microscope let scientists see the membrane in much greater detail. Studies of human red blood cells, RBCs, gave important clues since RBCs are simple cells with clear membranes. Chemical analysis showed membranes are made mostly of lipids and proteins. Major components of the membrane. 1. Lipids, mainly phospholipids. 2. Proteins. 3. Carbohydrates.
Cholesterol!
The lipid bilayer. The membrane's main structure is made of phospholipids arranged in a bilayer, 2 layers of molecules. Each phospholipid has a hydrophilic water-loving head containing phosphate that faces water. 2 hydrophobic water-hating tails made of fatty acids that avoid water. In the bilayer, the heads face outward toward watery environments inside and outside the cell. The tails face inward, hiding from water by pointing at each other. This arrangement creates a stable barrier between the cell's watery inside and outside. Why the bilayer arrangement is important. The hydrophobic core, tails, blocks most water-soluble substances like ions and sugars from freely passing through. This controls what enters or exits the cell, helping the cell maintain a stable internal environment. Homeostasis. Only small non-polar molecules like oxygen or carbon dioxide can easily pass through the lipid bilayer. Role of cholesterol. Cholesterol molecules insert themselves between phospholipids in the bilayer. They make the membrane less rigid in cold temperatures, preventing it from becoming brittle, and less fluid in hot temperatures, preventing it from becoming too runny. Cholesterol stabilizes the membrane's fluidity, allowing the cell to function across temperature changes. Membrane proteins. Proteins are embedded in or attached to the membrane, performing vital functions. Integral proteins are partly or completely buried in the lipid bilayer. Some form channels or pumps that let specific substances cross the membrane. Peripheral proteins sit on the membrane's surface, inside or outside, help with cell signaling, shape or anchoring parts of the cell. Types of membrane proteins and their functions. Transport proteins move substances in or out of the cell, like doors. Receptor proteins receive signals from hormones or other molecules outside the cell, like antennas. Enzymes speed up chemical reactions at the membrane. Anchor proteins connect the membrane to the cell's internal skeleton or to neighboring cells. Glycoproteins, proteins with sugars attached, act as cell ID tags for recognition by other cells. Carbohydrates in the membrane. Carbohydrates are attached to lipids, glycolipids or proteins, glycoproteins, on the outer surface of the membrane. They form the glycocalyx, a fuzzy-looking coating outside the cell. Functions. Cell recognition, like name tags that tell other cells who they are, important for the immune system. Cell adhesion helps cells stick together to form tissues. Protection acts as a barrier against physical and chemical damage. Different membranes, different composition. Not all cell membranes have the same ratio of lipids, proteins, and carbohydrates. Red blood cells, erythrocytes, 52% protein, 40% lipids, 8% carbohydrates. Cells with many transport or signaling roles, like nerve cells, have more proteins in their membranes. The fluid mosaic model. The fluid mosaic model, proposed by Singer and Nicholson in 1972, explains the membrane's structure. Fluid. The lipid bilayer behaves like a thick liquid. Lipids and proteins can move sideways, giving the membrane flexibility. Mosaic. Proteins, lipids, and carbohydrates are scattered like tiles in a mosaic art piece. This dynamic nature lets the membrane change shape when the cell moves or divides, repair itself if damaged, adjust to environmental changes. Why the cell membrane is vital. Maintains a stable internal environment by controlling what goes in and out. Allows communication with other cells via proteins and carbohydrates. Supports cell movement, shape, and connection to other cells. Protects the cell's internal parts from the outside world.
No.
One sentence deepest summary. The cell membrane is a flexible, self-repairing, double-layered structure of phospholipids, cholesterol, proteins and carbohydrates arranged in a fluid mosaic that creates a selectively permeable barrier around the cell, enabling it to control what enters and exits, interact with other cells, and maintain the stability necessary for life. Would you like me to make a detailed diagram, infographic, or animation based on this summary? Absolutely. Here's a great, deepest, detailed yet crystal clear summary of everything you need to know about the fluid mosaic model and transport across the cell membrane, combining the idea in the simplest words, but with deep detail so you understand not but also why and how. The cell membrane, the flexible gatekeeper of life. The cell membrane, or plasma membrane, is the thin, flexible layer surrounding every living cell. 2. Its main job is to protect the cell, control what goes in or out, and communicate with other cells. Fluid mosaic model, 1972. In 1972, scientists Singer and Nicholson described the cell membrane as a fluid mosaic model. Fluid. The membrane is like a thick moving liquid. Parts of it can slide around. Mosaic. It's made of many small pieces, phospholipids, cholesterol, and carbohydrates arranged like tiles in a mosaic artwork. The fluidity comes from the phospholipid bilayer, which is not rigid but can shift, bend, and repair. Structure of the membrane. Phospholipid bilayer, 2 layers of phospholipids with hydrophilic heads facing water outside and inside the cell. Hydrophobic tails face each other inside, avoiding water. Creates a selectively permeable barrier. Only some substances pass directly. Cholesterol sits among phospholipid, controls fluidity, prevents membrane from becoming too stiff in cold, prevents it from becoming too fluid in heat. Proteins. Integral proteins go through or deep into the membrane, act like doors, channels, or pumps. Peripheral proteins sit on the surface, help with signals, cell shape, or attaching to other cells. Carbohydrates, attached to lipids, glycolipids, or proteins, glycoproteins, stick out of the cell surface, work like name tags, help cells recognize each other, key for immune system. Why is fluidity important? Fluidity of the membrane allows the cell to grow, membrane stretches as cell gets bigger, divide, membrane pinches and seals when cell splits, form junctions, cells can stick together to build tissues, send and receive signals, proteins can move to meet other molecules, take in endocytosis or send out exocytosis substances, membrane can fold in or out. Selective permeability. The membrane lets some substances cross freely but blocks others. This keeps the inside of the cell balanced and healthy. This selective permeability makes the membrane like a smart security system, choosing what enters or exits. How molecules move across the membrane. 1. Passive transport. No energy needed. Simple diffusion. Molecules like oxygen, O2 or carbon dioxide, CO2, move directly through the membrane from high to low concentration. Happens naturally without help. Osmosis. Special diffusion of water through the membrane. Water moves from high water concentration, low water concentration, keeps the cell's water levels balanced. Facilitated diffusion. Molecules like glucose or ions that can't cross on their own use carrier proteins or channel proteins. Still from high to low concentration and no energy used, but needs a helper. HASH2, active transport, requires energy, ATP. Moves molecules against the concentration gradient from low to high concentration, like pushing a ball uphill. Needs ATP energy because it goes against the natural flow. Example, NRK pump. Pumps 3 sodium ions out and 2 potassium ions in for every ATP used. Keeps balance of ions, essential for nerve and muscle function. Why can't some molecules cross alone? Molecules that are too big, like proteins, polar or charged, like ions, glucose, these can't pass through the hydrophobic, water-hating lipid tails in the membrane. They need channel or carrier proteins, like tunnels or ferry boats. Examples to make it clear. Simple diffusion, dye spreading in water, happens naturally. Osmosis, raisins swell in water, water moves into them. Facilitated diffusion, glucose using a special channel to enter the cell. Active transport, sodium-potassium pump, pushing ions against their natural direction using ATP. The biggest takeaway: the cell membrane is a soft, flexible, moving barrier made of fats, proteins, cholesterol and sugars. Thanks to its fluid mosaic design, it can change shape, repair itself, select what comes in and goes out, help cells stick together, communicate with other cells. Transport happens in two main ways. Passive transport, no energy, moves with the flow from high to low concentration. Active transport needs energy, moves against the flow from low to high concentration. Final one-line summary. The fluid mosaic model describes the cell membrane as a dynamic, flexible, self-repairing, selectively permeable barrier that lets cells control what enters or leaves by passive or active transport, ensuring life can grow, communicate, and survive. Would you like a diagram, animation, or interactive quiz to reinforce this summary? Absolutely. Here's a great, deepest, most detailed yet crystal clear summary of everything about the cell wall, explained step by step, but in the simplest words, so you know exactly what it is, what it does, how it's built, and why it matters. What is the cell wall? The cell wall is a tough, rigid, non-living layer outside the plasma cell membrane in plants, fungi, algae, and some bacteria. It is not found in animal cells, which is why animal cells are soft and flexible. The cell wall's main job is to protect, support, and shape the cell. What does the cell wall do? Gives shape. It keeps plant cells in a strong, boxy shape so they don't collapse or burst. Provides strength. Like an armoured shield, it lets plants grow tall and withstand wind or rain without falling over. Protects the cell. Stops mechanical damage like being pressed, and infection, keeps out many bacteria, fungi. Controls large molecules, acts as a barrier so big harmful substances, bad macromolecules, can't enter the cell. Help cells stick together. So many plant cells can form firm tissues like stems, roots, leaves. Allows communication. Tiny tunnels called plasmodesmata let neighbouring cells share water, nutrients, and signals. What is the cell wall made of? In plants, mainly cellulose, strong fibres that make up paper and cotton. Hemicellulose helps bind cellulose fibres together. Pectins, jelly-like substances that give flexibility and stickiness. Proteins provide extra strength. In algae, cellulose, galactans and mannans, special sugars. Calcium carbonate, same mineral as in shells, gives extra hardness. In fungi, chitin, also found in crab shells and insect exoskeletons. How does the cell wall develop? Primary cell wall. This is the first wall formed when a plant cell is young. It's thin, flexible, stretchy, allowing the cell to grow bigger. Secondary cell wall. As the cell ages, a second, thicker, stronger wall grows inside the primary wall, closer to the plasma membrane. The secondary wall gives extra strength and rigidity, important in parts like wood. What is the middle lamella? The middle lamella is a sticky layer between plant cells. It's made mainly of calcium pectate, a jelly-like glue. It cements neighbouring plant cells together so they stay attached and form solid tissues. Without the middle lamella, plant cells would fall apart. Example. Think of bricks in a wall. The middle lamella is like the cement holding them together. What are plasmodesmata? Plasmodesmata, singular plasmodesma, are tiny tunnels that pass through the cell wall and middle lamella. They connect the insides of neighbouring cells, letting them share water, minerals and nutrients, chemical signals. Through plasmodesmata, cells can coordinate activities like growing or fighting disease. Example, like secret passageways between rooms in a castle, allowing messages or supplies to pass through. Why is the cell wall important? Strength lets plants grow tall without flopping over. Protection blocks many germs, damage. Shape maintains plant cells' structure even when they fill with water, turgor pressure. Plasmodesmata connect cells into a working community. Support for tissues sticks cells together using middle lamella, building organs like leaves, stems, and roots. Why don't animals have a cell wall? Animals need to move freely. A rigid wall would make them stiff and unable to bend. Animals have skeletons or exoskeletons to support and protect them instead. Their cells use a flexible membrane only, which lets them change shape, e.g. white blood cells squeezing through tiny spaces. Quick comparison with cell membrane. Cell membrane, thin, flexible, living layer controls what goes in and out, found in all cells, plant, bacteria, fungi. Thick, rigid, non-living layer gives shape and protection, found in plants, fungi, algae, some bacteria, not in animal cells. Final simple takeaway. The cell wall is a strong, non-living outer layer around plant, fungal, and algal cells. It's made of fibers like cellulose and sticky substances like pectin. It protects the cell, gives it shape, and glues cells together with the middle lamella. Tiny tunnels called plasmodesmata connect cells so they can share nutrients and messages. The cell wall is essential for building strong, tall plants and forming firm tissues, but animals don't have one because they need to move. Would you like a diagram, visual comic, or interactive quiz to check your understanding of the cell wall? Of course, here's a great, deep, detailed yet crystal clear summary of the endomembrane system, bringing together all the key points in the simplest words so you fully understand what it is, how it works, and why it matters.
That's a sensor.
The endomembrane system, the cell's coordinated delivery network. The endomembrane system is a team of connected organelles inside eukaryotic cells. It is called a system because these organelles work together. They make, modify, transport, store, or recycle important molecules inside the cell. What organelles are included? 1. Endoplasmic reticulum ER. Rough ER has ribosomes, makes proteins. Smooth ER makes lipids, fats, and helps detoxify harmful substances. ER sends these products to the Golgi. 2. Golgi apparatus, Golgi complex. Like a post office, receives proteins and lipids from the ER, modifies them, e.g. adds sugars, sorts them, packs them into vesicles, and sends them to their destinations inside or outside the cell. 3. Lysosomes, like garbage trucks, break down waste, damaged cell parts or molecules and recycle them, contain powerful digestive enzymes. 4. Vacuoles, like storage tanks, store water, nutrients or waste. In plants, the central vacuole maintains turgor pressure, keeping the plant upright. Organelles are not part of it. Mitochondria, chloroplasts, and peroxisomes are not part of the endomembrane system. Why? Because they work independently on jobs like energy production, mitochondria, photosynthesis, chloroplasts, or detoxifying chemicals, peroxisomes, without directly coordinating with the ER, Golgi, lysosomes, or vacuoles. Why do we call it a system? These organelles pass materials between each other, usually in vesicles, small bubbles of membrane. They use similar membrane structures. Vesicles can bud off one organelle and fuse with another seamlessly. Together, they form an assembly line where molecules made by the ER are processed by the Golgi, sent where needed, stored in vacuoles, or broken down by lysosomes. Key jobs of the endomembrane system. Makes proteins and lipids, essential for life. Modifies and sorts products so they work properly and reach the right place. Packages molecules into vesicles, like boxes for shipping. Transports materials inside the cell or to the cell surface for export. Stores substances in vacuoles until needed or until they can be disposed of. Recycles waste. Lysosomes break down old or damaged parts so the cell stays clean. Simple analogy. Imagine a factory. Is like the assembly line makes products. The Golgi is like the packaging and shipping department, prepares and directs products. Lysosomes are like the recycling unit, break down trash and reuse parts. Vacuoles are like the warehouse, store products until needed. All these parts must work together or the factory cell will stop functioning.
Big picture.
The endomembrane system allows the cell to produce, process, move and manage everything it needs, from proteins that build structures and enzymes, to lipids for membranes, to waste removal and storage. Without this system, the cell couldn't grow, repair or maintain itself. Final one-line summary. The endomembrane system is the cell's team of organelles, Golgi, lysosomes and vacuoles, that work together to make, process, transport, store and recycle important molecules, keeping the cell alive and organized. Would you like a diagram, animation or practice questions on the endomembrane system? Of course. Here's a great, deepest, most detailed yet crystal clear summary of everything about the endoplasmic reticulum, covering every important point so you fully understand what it is, how it works, and why it's so vital to cells. What is the endoplasmic reticulum, The endoplasmic reticulum, is a large, complex network of tubes and flattened sacs inside eukaryotic cells, plants, animals, fungi, protists, but not bacteria. The spreads through the cell's cytoplasm, reaching from the nuclear envelope, outer layer of the nucleus, out into the cell. Under an electron microscope, scientists see the as a maze-like structure. Two spaces created by the The divides the inside of the cell into one luminal, cisternal space, the hollow space inside the ER's tubes and sacs. Two, extraluminal space, the rest of the cytoplasm outside the This separation lets the cell control which molecules stay inside the for remain processing and which remain in the cytoplasm. Types of rough and smooth. The comes in two types based on whether ribosomes are attached. One, rough endoplasmic reticulum. Appearance looks rough under the microscope because of ribosomes, small dots, attached to its outer surface. Main job, makes proteins, especially those that will be secreted out of the cell, like hormones or enzymes, will become parts of the cell membrane, will go to lysosomes. How it works. Ribosomes on the RER read messenger RNA from the nucleus and build proteins by linking amino acids. Newly made proteins enter the RER lumen where they start folding into their correct shapes. Who has lots of RER? Cells that produce and secrete lots of proteins, e.g. pancreatic cells making insulin, immune cells making antibodies. Connection to nucleus. RER is directly continuous with the outer membrane of the nuclear envelope, allowing fast transfer of genetic instructions. 2. Smooth endoplasmic reticulum. Appearance looks smooth because it doesn't have ribosomes. Main jobs, makes lipids, fats, including phospholipids for new membranes and cholesterol. Makes steroid hormones in animal cells, e.g. estrogen, testosterone, cortisol. Detoxifies harmful chemicals, especially in liver cells. CR helps break down alcohol, drugs and poisons. Stores calcium ions in muscle cells. CR releases calcium needed for muscle contraction. Who has lots of SER? Cells specialized in making lipids or hormones, e.g. adrenal gland cells, liver cells, muscle cells. Rough and smooth work together. RER and CR are interconnected parts of the same continuous network, but they specialize in different jobs. RER, protein production. SCR, lipid production, detoxification and calcium storage. Together, they produce most of the molecules the cell needs to build and maintain itself. HAB, role in the endomembrane system. The is the first major station in the endomembrane system, a coordinated network of organelles, Golgi, lysosomes, vacuoles, that makes, processes and moves materials inside the cell. After proteins are made in the RAR, they are packaged into small transport vesicles. These vesicles bud off from the and travel to the Golgi apparatus for further modification, sorting and shipping. Importance of the to the cell. Builds proteins and lipids. essential for membranes, enzymes, hormones, and signaling molecules. Folds and modifies proteins, helps them work properly. Misfolded proteins can cause diseases. Detoxifies harmful substances, especially in liver cells. CR keeps the body safe. Stores calcium, critical for muscle contraction and cell signaling. The ER also maintains the cell's membrane supply, repairing damage and helping the cell grow.
NASCAR.
Simple analogy. Think of the ER as a massive factory inside the cell. RER, assembly line making proteins. CR, department making fats and detoxifying chemicals. The ER is connected to the control center nucleus and sends products to the shipping center, Golgi. Final summary. The endoplasmic reticulum, ER, is a large connected network of membranes inside eukaryotic cells.
RER.
Covered with ribosomes makes proteins. Smooth without ribosomes makes lipids, steroid hormones, detoxifies harmful substances, and stores calcium. The ER splits the cell's inside into luminal and extraluminal spaces, helps fold and process proteins, and connects directly to the nucleus. The ER is essential for building, organizing, and maintaining the cell. Without it, the cell couldn't survive. Would you like a detailed diagram, interactive quiz, or cartoon to help you remember the ER? Of course! Here's a great, deep, detailed summary, but still explained as simply as possible, so you get every important piece and nothing is missed. This will give you a full, crystal clear understanding of the Golgi apparatus. What is the Golgi apparatus? The Golgi apparatus, also called the Golgi body or Golgi complex, is an organelle, tiny machine inside eukaryotic cells, cells with a nucleus, discovered by Camillo Golgi in 1898 when he saw it as darkly stained structures near the nucleus. Structure of the Golgi, made of many flat disc-like sacs called cisterna, singular cisterna. These cisternae are stacked one on top of the other, like a pile of pancakes or plates. Each cisterna is 0.1.0 micrometer wide, extremely small. The whole stack looks a bit curved and is polarized. It has a definite direction with a cis face and a trans face.
Yeah.
This face, forming face, convex, bulges outward, located near the endoplasmic reticulum ESR, receives materials like proteins and lipids from the ER via small transport bubbles called vesicles. Trans face, maturing face, concave, curves inward, faces toward the cell membrane, packages finished products into vesicles and sends them to their destinations inside the cell or outside. These two faces are functionally and structurally different, but connected by the middle cisternae, which help materials move step by step from cis to trans. How does the Golgi work? 1. Receiving materials. Proteins and lipids made in the rough or smooth ER are packed into vesicles. These vesicles fuse with the cis face of the Golgi. 2. Processing and modifying materials. As proteins and lipids move from one cisterna to the next, cis, medial, trans, they are chemically modified. Examples of modifications: adding sugar chains to proteins makes glycoproteins. Adding sugars to lipids makes glycolipids. Cutting or folding proteins into their final shape. 3. Sorting and packaging materials. The Golgi sorts these products, adds molecular labels like postal addresses, and packs them into new vesicles at the trans face. Different vesicles go to different places. Some go to the cell membrane to release Of course. Here's a great, deep, detailed summary of lysosomes, still in simple, clear words, but covering every important point you need to truly understand them. What are lysosomes? Lysosomes are small, membrane-bound sacs found inside almost all animal cells. They are filled with digestive enzymes, proteins that break things down. Their main job is to digest or break down unwanted materials inside the cell. How are lysosomes made? Lysosomes are formed by the Golgi apparatus, which packages enzymes into small vesicles. These enzyme-filled vesicles bud off the Golgi. These vesicles become lysosomes. Simple picture. Golgi equals factory, fills bags with enzymes, bags equals lysosomes. What is inside a lysosome? Lysosomes contain many hydrolytic enzymes, enzymes that cut molecules by adding water. These enzymes are called hydrolases, and they come in many types. Lipases break down fats, lipids. Proteases break down proteins. Carbohydrases break down carbohydrates, sugars, starches. Nucleases break down nucleic acids, DNA and RNA. These enzymes can digest almost any kind of biomolecule the cell needs to get rid of. Why are lysosomes acidic? The inside of lysosomes is acidic with a pH around 4.5-5.0, similar to lemon juice. This acidic environment helps the enzymes work best. The lysosome membrane has special proteins that pump protons, H' ions, into the lysosome, keeping it acidic. Simple example, like your stomach needs acid to digest food, lysosomes need acid to digest materials inside the cell. What do lysosomes do? Digest worn-out cell parts, autophagy. When parts of the cell, like old mitochondria, stop working, lysosomes break them down and recycle their materials. Break down materials taken in from outside the cell, heterophagy. When a cell eats bacteria or particles, lysosomes fuse with these vesicles and destroy the contents. Recycle building blocks. After digestion, the small pieces, amino acids, sugars, fats, can be reused by the cell to build new molecules. Destroy harmful invaders. Lysosomes help the immune system by breaking down bacteria or viruses that get inside the cell. How does digestion happen? 1. Material to be digested, damaged organelle, bacteria, etc., is enclosed in a bubble called a vesicle. 2. This vesicle fuses with a lysosome. 3. Lysosome enzymes digest the contents, breaking them into smaller reusable parts. 4. Useful parts go back to the cell. Waste parts may be released outside the cell. Why is the membrane important? The lysosome's membrane keeps the powerful enzymes inside. If the membrane breaks, the enzymes can spill out and damage or kill the cell by digesting its own parts. The membrane also protects the rest of the cell from the acid inside the lysosome. What happens if lysosomes don't work? If lysosomes don't have working enzymes because of genetic problems, the cell can't break down certain materials. These materials build up inside the cell and can cause diseases called lysosomal storage disorders. Examples of such diseases include Tay-Sachs disease, Gaucher disease, Pompe disease. Other important functions. Lysosomes are involved in programmed cell death, apoptosis, a normal process where cells self-destruct when they are old or damaged. Lysosomes can release their enzymes outside the cell to help break down materials in the space between cells, important in some immune responses. Recap. Why are lysosomes important? They keep the cell clean by removing and recycling waste. They help protect the cell by digesting harmful invaders. They allow the cell to reuse valuable building blocks, saving energy and resources. Without lysosomes, cells would get clogged with waste and stop functioning properly, leading to disease or death. Super simple, big picture. Lysosome equals cell's garbage truck, recycling center, plus defense system. Picks up and digests old, damaged parts or invaders, recycles useful parts, keeps the cell safe and clean. Keywords to remember. Lysosome, membrane-bound sac of enzymes. Enzymes, hydrolases, cut up big molecules. Acidic pH helps enzymes work. Autophagy, digesting own worn-out parts. Heterophagy, digesting things from outside like bacteria. Golgi apparatus makes lysosomes. Would you like a simple diagram or a step-by-step animation script to visualize lysosomes? Absolutely. Here's your great detailed summary of mitochondria, explained clearly and simply, but covering everything important you need to know. What are mitochondria? Mitochondria, singular mitochondrion, are tiny sausage or tube-shaped structures inside almost all your cells. They are called powerhouses of the cell because they make energy. Why are they called powerhouses? Mitochondria use oxygen to break down food, glucose and fats and make ATP, adenosine triphosphate. ATP is the cell's main energy currency. It powers everything the cell does, moving, growing, dividing, transporting molecules and more. Without mitochondria, cells would not have enough energy to survive. How many mitochondria are in a cell? The number of mitochondria varies greatly depending on how much energy the cell needs. Cells that use lots of energy, like muscle or nerve cells, have thousands of mitochondria. Cells that need little energy have fewer mitochondria. What do mitochondria look like? They are usually sausage-shaped or cylindrical, but their shape can change. They range from 0.2 to 1.0 micrometers in diameter and 1.0 to 4.1 micrometers in length. Very tiny, only visible with a microscope. What are mitochondria made of? Each mitochondrion has two membranes. Outer membrane, smooth outer covering. It allows small molecules and ions to pass easily. Inner membrane, folded into structures called cristae. 2 cristae, the folds of the inner membrane that stick into the inside of the mitochondrion. They increase the surface area, giving more space for energy-making reactions. 3 matrix, the jelly-like inside of the mitochondrion enclosed by the inner membrane, contains enzymes, mitochondrial DNA, RNA, ribosomes, and everything needed for the mitochondrion's own protein synthesis. Why are cristae important? Cristae increase the surface area of the inner membrane. This means more space for the proteins and enzymes involved in making ATP, so mitochondria can produce more energy quickly. What happens in mitochondria? Mitochondria carry out aerobic respiration, a multi-step process that includes glycolysis, breaks down glucose outside mitochondria in the cytoplasm. Krebs cycle, citric acid cycle, takes place in the matrix, breaks down molecules further, releasing energy. Electron transport chain, ETC, happens on the cristae. Electrons from food move through proteins, pumping protons and creating a gradient that makes ATP. Oxygen is used in the final step of the ETC, where it combines with electrons and protons to make water. That's why we breathe oxygen. What do mitochondria make? They produce ATP, up to 38 ATP molecules per glucose molecule burned. ATP stores energy in a form cells can use immediately. What special features do mitochondria have? Own DNA. Mitochondria contain circular DNA separate from the cell's main DNA in the nucleus. Their DNA codes for some of the proteins they need to function. Own ribosomes. Mitochondria have small ribosomes, 70S type, like bacteria, to make some of their own proteins. Multiply by fission. Like bacteria, mitochondria reproduce by splitting into 2, binary fission. When a cell needs more energy, mitochondria can divide to increase their number. Of course, here's your great, deep, detailed summary of plastids in clear, simple language, but covering every important detail so you get the complete picture. What are plastids? Plastids are large membrane-bound structures found inside all plant cells and euglenoids, plant-like single-celled organisms. They are easily seen under a microscope because they're bigger than many other cell parts. Plastids are unique to plants and certain algae. Animal cells do not have plastids. Why are plastids important? Plastids give plants their colors, make food, or store nutrients. They contain pigments, colored molecules, or store materials like starch, oils, or proteins. Plastids are essential for photosynthesis, making food with sunlight, storing energy, attracting pollinators with bright colors, helping seeds grow by providing stored nutrients. Types of plastids. Plastids are classified into 3 main types depending on their pigments or stored materials. 1. Chloroplasts, green food factories, contain chlorophyll, the green pigment that absorbs sunlight. Also have carotenoids, but chlorophyll's green color dominates. Chloroplasts trap light energy and use it to perform photosynthesis. converting sunlight, water, and carbon dioxide into glucose, sugar, and oxygen. Chloroplasts are made of double membrane, outer and inner membranes, grana, stacks of membranes, thylakoids, where light energy is captured, stroma, the fluid-filled space inside the chloroplast where sugar is produced. Chloroplasts give plants their green color. They are found mainly in leaves and green stems. Why chloroplasts matter? They make almost all the food for life on Earth and produce the oxygen we breathe. 2. Chromoplasts. Color painters contain carotenoid pigments such as carotene orange, xanthophylls, yellow, other red pigments. Chromoplasts give yellow, orange, or red colors to parts of the plant like flowers to attract pollinators, fruits to attract animals for seed dispersal. Some roots, e.g. carrots, storage organs, chromoplasts form when chloroplasts change during ripening. Example, a green tomato has chloroplasts. As it ripens, chloroplasts turn into chromoplasts. The tomato turns red. Why chromoplasts matter? They help plants reproduce by attracting animals and insects. 3. Leucoplasts. Colorless storerooms, no pigments, they are white or colorless. Found in plant parts not exposed to light, like roots, tubers, seeds, and underground stems. Main job, store nutrients to help the plant survive or help seeds germinate. Types of leucoplasts. Amyloplasts store starch, a carbohydrate, found in potatoes, rice, wheat, and other storage organs. Amyloplasts give potatoes their white, starchy texture. Elaioplasts store oils and fats found in seeds like sunflower or castor oil seeds, provide energy for germination or seedling growth. Aleuroplasts store proteins found in protein-rich seeds like beans or peas, provide building blocks for growing seedlings. How do plastids form? Plastids come from proplastids, small, undifferentiated plastids found in young plant cells. Depending on the plant's needs, proplastids can develop into chloroplasts if the cell needs to make food with light, chromoplasts if the cell needs bright color, e.g. fruit ripening, leucoplasts if the cell needs to store nutrients. Example, in a green tomato, chloroplasts make it green. When the tomato ripens, chloroplasts turn into chromoplasts. Tomato turns red. Why do plants need different plastids?
Yeah.
Chloroplasts make food, keep the plant alive. Chromoplasts make bright colors, help pollination or seed dispersal. Leucoplasts store food, help plants survive tough times and help seeds grow. Plastid pigments, the key to color and energy. Pigments in plastids absorb light and reflect certain colors. Chlorophyll absorbs red-blue, reflects green-green leaves. Carotenoids absorb blue-green, reflect yellow, orange, red, colored flowers, fruits. No pigments, leucoplasts look white or clear. Summary in one simple line. Plastids are plant cell parts that make food, give colors, or store food, helping plants grow, survive, and reproduce. Key points to remember. Plastids equals unique to plants, euglenoids. Plastids are large, easy to see with a microscope. Types chloroplasts, green, photosynthesis. Chromoplasts, yellow, orange, red, attract animals, insects. Leucoplasts, colorless, store starch, fats, or proteins. Plastids develop from proplastids, depending on what the plant needs. Plastids' pigments decide plants' colors and help them make or store food. Would you like a diagram or visual guide showing the 3 plastid types side by side? Of course, here's your great, deep, detailed summary of plastids in clear, simple language, but covering every important detail so you get the complete picture. What are plastids? Plastids are large membrane-bound structures found inside all plant cells and euglenoids, plant-like single-celled organisms. They are easily seen under a microscope because they're bigger than many other cell parts. Plastids are unique to plants and certain algae. Animal cells do not have plastids. Why are plastids important? Plastids give plants their colors, make food, or store nutrients. They contain pigments, coloured molecules, or store materials like starch, oils, or proteins. Plastids are essential for photosynthesis, making food with sunlight, storing energy, attracting pollinators with bright colours, helping seeds grow by providing stored nutrients. Types of plastids. Plastids are classified into three main types depending on their pigments or stored materials. 1. Chloroplasts. Green food factories contain chlorophyll, the green pigment that absorbs sunlight. Also have carotenoids, but chlorophyll's green colour dominates. Chloroplasts trap light energy and use it to form photosynthesis, converting sunlight, water and carbon dioxide into glucose, sugar and oxygen. Chloroplasts are made of double membrane, outer and inner membranes, grana, stacks of membranes, thylakoids, where light energy is captured, stroma, the fluid-filled space inside the chloroplast where sugar is produced. Chloroplasts give plants their green colour. They are found mainly in leaves and green stems. Why chloroplasts matter? They make almost all the food for life on Earth and produce the oxygen we breathe. 2. Chromoplasts. Colour painters contain carotenoid pigments such as carotene orange, xanthophylls, yellow, other red pigments. Chromoplasts give yellow, orange or red colours to parts of the plant like flowers to attract pollinators, fruits to attract animals for seed dispersal. Some roots, e.g. carrots, storage organs, chromoplasts form when chloroplasts change during ripening. Example, a green tomato has chloroplasts. As it ripens, chloroplasts turn into chromoplasts. The tomato turns red. Why chromoplasts matter? They help plants reproduce by attracting animals and insects. 3. Leucoplasts. Colourless storerooms, no pigments, they are white or colourless. Found in plant parts not exposed to light, like roots, tubers, seeds and underground stems. Main job, store nutrients to help the plant survive or help seeds germinate. Types of leucoplasts. Amyloplasts store starch, a carbohydrate, found in potatoes, rice, wheat and other storage organs. Amyloplasts give potatoes their white starchy texture. Elaioplasts store oils and fats found in seeds like sunflower or castor oil seeds, provide energy for germination or seedling growth. Aleuroplasts store proteins found in protein-rich seeds like beans or peas, provide building blocks for growing seedlings. How do plastids form? Plastids come from proplastids, small, undifferentiated plastids found in young plant cells. Depending on the plant's needs, proplastids can develop into chloroplasts if the cell needs to make food with light, chromoplasts if the cell needs bright colour, e.g. fruit ripening, leucoplasts if the cell needs to store nutrients. Example, in a green tomato, chloroplasts make it green. When the tomato ripens, chloroplasts turn into chromoplasts. Tomato turns red. Why do plants need different plastids?
Yeah.
Chloroplasts make food, keep the plant alive. Chromoplasts make bright colours, help pollination or seed dispersal. Leucoplasts store food, help plants survive tough times and help seeds grow. Plastid pigments, the key to colour and energy. Pigments in plastids absorb light and reflect certain colours. Chlorophyll absorbs red-blue, reflects green-green leaves. Carotenoids absorb blue-green, reflect yellow, orange, red, coloured flowers, fruits. No pigments, leucoplasts look white or clear. Summary in one simple line. Plastids are plant cell parts that make food, give colours or store food, helping plants grow, survive and reproduce. Key points to remember. Plastids equals unique to plants, euglenoids. Plastids are large, easy to see with a microscope. Types chloroplasts, green photosynthesis. Chromoplasts, yellow, orange, red, attract animals, insects. Leucoplasts, colourless, store starch, fats or proteins. Plastids develop from proplastids, depending on what the plant needs. Plastids' pigments decide plants' colours and help them make or store food. Would you like a diagram or visual guide showing the three plastid types side by side? Absolutely. Here's your great deep detailed summary of ribosomes. in the simplest words but covering every important point so you get the complete picture. What are ribosomes? Ribosomes are tiny machines inside every living cell that make proteins. Proteins are needed for everything, building cell parts, enzymes, hormones, muscles, skin, hair, and more. Ribosomes look like small dots or grains when seen under a strong microscope. Who first observed ribosomes? George Palade, a scientist, first clearly saw ribosomes in 1953 using an electron microscope, a very powerful microscope that can see objects much smaller than light microscopes can. What are ribosomes made of? Ribosomes are built from two main things. Ribonucleic acid, RNA, reads instructions for making proteins. Proteins form part of the ribosome structure and help it work. Ribosomes do not have a membrane. They are open structures so they can grab RNA and start making proteins quickly. Where are ribosomes found? Ribosomes can be free in the cell's cytoplasm. Cell's jelly make proteins for use inside the cell. Attached to the rough endoplasmic reticulum, RER, make proteins that are sent outside the cell or used in the cell's membrane. That's why the RER looks rough under the microscope, because it's covered with ribosomes. Types of ribosomes, 80S versus 70S. Ribosomes come in two types based on the kind of cell. 80S ribosomes, found in eukaryotic cells, plants, animals, fungi, protists, cells with a nucleus. 70S ribosomes, found in prokaryotic cells, bacteria, archaea, cells without a nucleus. Ribosomes have two subunits. Every ribosome is made of two parts called subunits, like a hamburger bun. A large subunit, top bun, a small subunit, bottom bun. They come together when making a protein separate when not needed. Ribosome subunit sizes. In 80S ribosomes, eukaryotic, large subunit is 60S, small subunit is 40S. In 70S ribosomes, prokaryotic, large subunit is 50S, small subunit is 30S. Note, 60S plus 40S to 100S and 50S plus 30S to 80S, because S numbers don't add normally. What does S mean? S stands for Svedberg's unit, named after scientist Theodore Svedberg. It measures the sedimentation coefficient, how fast something sinks when spun at very high speeds in a centrifuge. Bigger and denser particles sink faster, have higher S values. Example, a rock sinks faster in water than a feather, rock would have a higher S. What do ribosomes do? Ribosomes read the mRNA instructions, messenger RNA, the copy of DNA's information, use them to link amino acids into long chains. These chains fold into proteins. Without ribosomes, a cell cannot make proteins. Life would not exist. Ribosomes are everywhere. Ribosomes are in all living things, plants, animals, bacteria, fungi, protists. Because all life needs proteins, ribosomes are universal and essential. Big picture summary. Ribosomes are tiny, essential machines inside all cells. They build proteins by reading instructions from RNA. First seen by George Palade in 1953 using an electron microscope made of RNA plus proteins, no membrane, allows two parts, large plus small subunits, and eukaryotic 80S ribosomes 60S plus 40S, prokaryotic 70S ribosomes or 50S plus 30S. S, Svedberg's unit, measures how fast ribosomes sink when spun, shows size density, not regular addition. Ribosomes can be in the cytoplasm or attached to rough ER, depending on where the protein is needed. Key takeaway, without ribosomes, no proteins, no life. Would you like a comic strip or diagram showing how ribosomes read RNA and build proteins?